Very nice. Very nice. Very warm welcome. So, thanks for coming. I really like to talk about fungi, and I'm happy to be here. This talk is about entomopathogenic fungi, hence the title here. And I was just flipping through this book. I have a few books that people can come up and take a look at after, or, well, maybe not during, but afterwards, and a lot of specimens that I've collected that you can also take a look at and poke your finger at if you want to. A couple of these books are from the 1800s, so this book is from 1893, and people had like a really different perception of fungi in 1893, which is not that long ago. They thought of them as plants, and they were not differentiated into like different families at all. So like what we're exploring today is like a really niche part of a niche study, and that's why I'm really excited about it, because like anyone can do it, especially with like the advent of like iNaturalist and community science and clubs like this. Yeah, I'm super excited to share it with you, so I'll just read you a little bit about how these fungi were perceived in 1893 by Julia P. Ballard. And yeah, no, I'm just going to do it. Among the most curious productions of New Zealand is a singular plant called by the natives aweto, the Sphoria robertia, or bulrush caterpillar. If nature ever takes revenges, one might imagine this to be a case of retaliation. Caterpillars live upon plants, devouring not only leaves but bark, fruit, pith, root, and seeds. In short, every form of vegetable life is drawn upon by these voracious robbers. And here come a little seed that seems to say, "Turn about is fair play," and lodges on the wrinkled neck of the caterpillar, just at the time when he, satisfied with his thefts of the vegetable kingdom, goes out of sight to change into a chrysalis and sleep his way into a new dress and a new life. A vain hope. The seed has the situation. It sends forth its tiny green stem, draws its life from the caterpillar, and not only sends up its little shoot with the bulrush stem capped with a tiny cattail, but fills with its root the entire body of its victim, changing it into a white pith-like vegetable substance. This, however, preserves the exact shape of the caterpillar. It is nut-like in substance and is eaten by the natives with great relish. A friend who has recently spent some months in New Zealand brought me the specimen, a drawing of which is here shown. There are other cases of this vegetable retaliation, but none so curious as this of the bulrush caterpillar. I thought it was hilarious, well written, and just like, I don't know, fascinating. Because like they understand the whole mechanism. They've like studied it very closely. They've looked at the spores and considered it a seed. We'll get into it a little bit, but feel free to like flip through a few of these books carefully. At the end, the illustrations are incredible because they couldn't take photos of these things, and illustrators were just like, scientific illustrators were like really talented in those days, and still are. But I think there was a different necessity that came with being an illustrator. So, who am I? I'm just some guy that likes to poke around under rocks, seen here in the wild. I was born in Durham, North Carolina, and I moved to Kennett Square to study horticulture. I'm a horticulture student at Longwood Gardens, and I graduate in July. And the first entomopathogenic fungus I ever found, and really like, this was around the same time that I was getting into mycology, was only about, it was August 1st, 2021, so it really wasn't that long ago. I find mycology to be like a really reciprocal scientific practice. Like if you're interested in something, generally people are down to answer your questions, and there are very few barriers to entry that might exist like within the walls of like an academic driven science where you have to go to school to study it, or I don't know, there's a lot of economic barriers to entry or other things to consider that a lot of other science a lot of other sciences have to deal with, but I think mycology has been kind of like shoved under the rug for so long and like thought of as this like thing that freaks do and like in the back of the, you know. So I'm really excited about that. I'm grateful to be a freak and a community scientist. What's community science? It's when people without like a traditional scientific background or with a scientific background get together with their community and just go out and do science. You know, this is like the age of accessible science. Everybody has like a smartphone pretty much. Everyone has a camera capable of capturing the world around them. And with the age of information that we're in, sharing those photos and identifying them and communicating about them has become so much easier. And it's not really just like a practice of the bourgeoisie or the wealthy, as it has been for hundreds of years, thousands of years. What is an entomopathogenic fungus? Entomopathogenic fungi are fungi that parasitize insects, typically resulting in their death. I'll show you one that classifies as an entomopathogenic fungus today on a ladybug, but it's kind of an STD and it just sort of impairs them. It doesn't kill them, to my knowledge. We'll talk about that a little bit later. Many species are host-specific, which is really exciting. What that means is that these host insects, so let's say carpenter ants. Carpenter ants are a huge group of ants. There's, I don't know how many species of carpenter ant, probably thousands, probably more to be discovered. There is a theory that people who study Ophiocordyceps, which is the genus of entomopathogenic fungus that infects carpenter ants, there's a theory going around that each fungus is host-specific to a specific species of carpenter ant. And that's supported by every species that's been discovered so far. And what that means is that these fungi have co-evolved alongside their hosts for thousands of years, and they play a role in sort of like the balancing act of ecology, the cycling of nutrients, because back to ants, the biomass of ants on this planet is like massive. Like there's so many ants, there's more ants, forget how to say this, but it's like there's more ants in weight on the planet than there are humans, is like the generalized statistic, which when you think about it is scary. But we're just learning now that, you know, in North Carolina, when I first discovered my first entomopathogenic fungus, it was on a beech tree. I was on a hike with my dad, and he pulled a twig off of a beech tree, like at eye level, and it had 4 infected ants on it. And then we looked at every other beech tree, like at our house, at the park down the road, like 20 miles away. Every single beech tree had like 60 to 80 ants on it, infected with this fungus. So think about like the nutrients that are being cycled. You know, this passive predator is infecting this insect that takes up such a vast amount of space and nutrients just by existing in the environment, and it's sort of predating that insect and turning those nutrients and churning them back into the environment. So I think it's really interesting. I'd like people to study it. That's why I talk about it. I just like to find them. I'm really interested mostly in the field work and the photography of these insects and these fungi. But I also think they're really beautiful. They're individual and expressive. That's why I like to photograph them. Most of this presentation is photographs, so I hope you can see all the way in the back. Because there's thousands of people here. You wouldn't know it from the recording, but just echoing space, you know. So a few terms that I will say, if you're not familiar with them, are genus. Maybe I've already said these terms. It's a taxonomic category that ranks above species and below family. Species is a taxonomic category below genus. So when I said Ophiocordyceps earlier, that infects the carpenter ants, I'm referring to a genus of fungi. And Ophiocordyceps campanoti pennsylvanicus is a new species of entomopathogenic fungus that we are in the process of describing, and from the Susquehanna, and that is an example of the species. A complex, which I will reference in this presentation, is a group of organisms that are so visually similar, the boundaries between them are unclear, so more work needs to be done to delineate them from a single species into new species, or maybe even some of them into a new genus. And morphology is just like the physical characteristics of an organism. Yeah. So, habitat. When I was running around in the woods in North Carolina with my dad, we scooped them up and we were flabbergasted by them, and we were like, this is a new discovery to science, this is incredible. And it was incredible, but it wasn't new to science. I just hadn't heard of them before. I'd watched this fantastic fungi. I'd read all the major media publications about zombie ants, and I was interested in them, but I didn't really understand their ecology, and I certainly didn't think they were in North America. So when I found them, I was like, this has got to be super rare. Then we started finding them everywhere, and I was like, well, if I come up to if I go back to PA from North Carolina, that's like a 6-hour drive north. The forests are pretty much the same. They're temperate deciduous forests. They have American beech trees. They have tulip poplars. So I imagine they have a lot of the same species of ants. I can probably find this ant in PA. I came up here, looked all over the beech trees, couldn't find a single thing for like a year. And while looking, I found a lot of Gibellula, which is an entomopathogenic fungus that infects spiders. I found beetles infected, moths infected. I started to branch out into new genera and new species and realized that it was really like a bigger part of ecology than I had realized. I got more into mycology that way and started to just notice more about my environment in general. But when I started to really try and hone in and focus on entomopathogens, because I wanted to know the width of species that were present in North America, or at least in Pennsylvania, or the southeast or northeast, whatever you want to say. I realized that I would find these pockets of humidity, really, in the summer around the Susquehanna River, because you have all these tributaries feeding down into the Susquehanna, and they're just tropical humidity in the summer. It's so hot, it's so wet all the time. And if you think about it, it's the perfect place for fungal spores to germinate. It's just like a humidity chamber. It is a humidity chamber. Drainage down into a confluence of water, that is the perfect habitat to look for these things. If you're in a more tropical region of the world, they're going to be everywhere. Depending on the time of year, but I mean, I'm very excited to try and get to Brazil, or I'm excited to explore the rainforest of the southern Appalachians, which is Nantahala. It's in North Carolina. It's where the Appalachian mountain range, all of the water empties into this little spot, and in the summer the humidity is similar to these little pockets along the Susquehanna, but it's 150,000 acres of forest, so I'm sure the species there are just super, super diverse, because the insect life is diverse. All of these little ecosystems that pop up are really diverse. So yeah, right now I'm studying horticulture in PA. That's why I've been focused in on PA, but every time I go to another state or visit a friend, I'm always going down in the creek and seeing what I can find. And the diversity regionally is super variable. It's amazing what you can find in Connecticut comparatively to PA, and North Carolina comparatively to New York. I mean, there's a lot of overlap, of course, but then you get different species of insect, and because they're host-specific fungi, you know, you have the opportunity to discover something in your backyard, or your friend's backyard, or, you know, any state you may go to. Think about, if you're looking for habitat, if you're thinking, oh, is it the right time to go out and look for these fungi? It's like any other fungus. You know, you're looking for morels, there's a time frame, there's a window that you should be doing that. It fluctuates now based on climate change, you know. So that's a factor to consider now as well. But you can think about the light intensity, the rainfall, all of these things are going to influence fungal germination. And yeah, the hosts are going to be, another sensationalized species is cordyceps, and people kind of colloquially use cordyceps as like a blanket term to refer to entomopathogenic fungus, so they don't have to say entomopathogenic fungus, like I've been like spitting out this entire time like an insane person. Maybe I'll refer to them as entos. That's a good one. And yeah, there's a wide variety of host insects. You can come up here at the end, and I will pass around this box, and you can see some of the ones that I've collected in the region. But I mean, Lepidoptera pupa, which are moths and butterflies in their developmental stage in the summer, are really common. Gibellula in the fall because of beech leaf senescence. So beech trees, American beech trees, are the last trees to lose their leaves. And if you walk through the forest in the fall and look up, there's no overstory canopy. So this understory tree, you can see the light shining straight through the leaves. And it's really beautiful to just walk through, and it's kind of like a meditative practice of mine. But I was just doing that, and I noticed that you can see every little insect that, or spider, or whatever it may be, through these leaves. And it's a really, really good way to find spiders infected with Gibellula. In the fall, you can pretty much go anywhere and find Gibellula. It's a very common species. It's really charismatic. It's really beautiful. We'll look at photos of it. I don't know what I meant by learning to see. Maybe I was learning to see ecology. I don't know. It really helped me, I guess, like studying entos has helped me conceptualize life and death as a young person into something different than what society tells us, like, as humans, life and death is. And of course, like, those things are true. Like, if I were to lose a loved one, I would be very upset and it would hurt me. But I think it's helped me process a lot of death in my life, because I get to go out and see these fungi just working on the ecosystem, and sort of life and death churning up the woods and the earth, and it's kind of just like one continuous thing rather than this black and white event. Cordyceps militaris, everyone's favorite. The hosts include emerging cicada larvae, lepidopteran larvae, which is what I most commonly find it on, although here is actually that's a Lepidoptera pupa, probably like an oak moth. Never mind, thought it was a cicada. Just kidding. They're commonly found in early to late summer in mossy areas, and some places like in upstate New York, people are just like finding them everywhere. I've only ever found 2. And I know people that have just found like hundreds every year. I mean, I think that says more about the habitat where they're found and like the host being in that habitat than it does about, like, I don't know, my ability to find them or something. I've been looking hard, and this is like really the only one I've ever found. I've been at forays where they were found mostly in the Poconos and in upstate New York. But it's really beautiful, and if you get up close, you can see all of the little spore heads ready to shoot off. And I got down close to photograph this, and as I looked through the camera, I could see spores being shot off, and they're like this long string just like being shot into the world. It's pretty amazing. Cordyceps tenuipes is, I'll say, the most common species that I find here in Pennsylvania. And I think its main host, although I would love to do DNA sequencing on the hosts that I'm finding, which would really like up my understanding of what's going on in the ecosystem. But I believe most of them are Lepidoptera larvae and pupa. Yeah, they're super charismatic. They're easy to spot, although they look a lot like bird poop, which a lot of these do. So spend a lot of time poking around under rocks, like touching bird poop. That's sort of like one of the one of the things you got to do if you want to study these. You're going to be touching a lot of bird poop. And yeah, there's that place in Kelly's Run. If anyone wants to go this summer, please let me know. Would love to go, because 1 or 10 mycologists, or 2 or 10 mycologists, always better than 1. Yeah, this place it's really special because you're walking through this creek system, and it goes down like 100 feet in elevation. It decreases, and the rocky like outcroppings beside you are just covered in moss, and you can just look through all of this moss. You can find ants infected with fungus. You can find spiders infected with fungus. Tons of these. I mean, and then you can find Bavaria bassiana infecting like the spotted lanternfly, which is pretty cool because that's like a invasive species being controlled by an endemic fungus. It is interesting though because it's not host specific, Bavaria bassiana, so it can infect like a lot of other coleoptera, a lot of other beetles and stuff. So I'm not sure if it's a good thing necessarily that these insects that are suddenly everywhere, the spotted lanternflies, are getting like epizootically infected. We'll say, yeah, by this fungus because it's just like spreading prolifically through the environment as a result. Yeah, one of the funny things of introduced species and invasives. You know, it's always like a tricky thing to try and figure out, but time will tell whether it's a problem or not. Acanthomyces arnerum. This is really cool. I have here, if you see one, there's always another, and I found that to be true. Like the photo of me like looking up under the rock, there's a creek running just to the left here, and there were some flies under here infected with a fungus, but then there was a spider web running like right beside me, and as I looked over I saw this, and this spider, along with the fungus infecting it, is like this big. It's like an inch. It's really, really big comparatively to like a lot of the other species or specimens that I'm finding, and it was suspended in its web. No, not a fan. It was like mummified though. It was really interesting because a lot of these have summit syndrome. Like a lot of the ants that get infected or the millipedes that get infected have summit syndrome, and they're climbing to a point where the fungus can then disperse the spores like the furthest. So I found it really interesting that in these enclosed spaces, that's the method of spore dispersal, is to like hang into like any wind that might be passing through these caves. And as a result, like this whole little community of spiders becomes infected. So there will be like 3 to 5 spiders in this cave all the time because that's like what the space allows, and they'll all be infected. And then over time, those spores will remain in the environment, but the spiders' bodies will like fall, and like the spider webs will erode away, and more spiders will move in. But the spores are in the environment already. So if they get them on them as they crawl through it, the whole cycle starts again. Acanthamyces tuberculatus is what I have here. And anybody off the top of their head know the complex name, Acanthamyces? It's not Arenarium. Okay. Because Acanthamyces tuberculatus, I think, was broken off as its own species. And there's another one that starts with an A that is now the complex. But this one is pretty inexplicable. Like a lot of the others, I find them in a habitat. I'm like, okay, this is where the insect lives. It makes sense. I'm going to explore this habitat next time I come upon it. Found it. It's just like, I'm in the woods randomly. There's a twig. It has one on it. It's completely just serendipitous. I have no idea what the host is. Obviously, it's an adult moth, which is really cool. You know, it like myceliates pretty like tight to the body of the moth, and then has all of these little growths coming off, which is really unique. Like this shot here is of the eye of the moth, and it's just totally covered in mycelium. But it's not like a glob or something. Like I find it really interesting that it maintains the shape. But I have no idea, like, are these nocturnal moths that are flying really low at night and somehow getting the spores on them? I don't know. I don't fully understand the spore dispersal method because they don't really look like fertile sporangia to me. But I don't know, I haven't looked into it. I'm interested to know. If anybody has any comments out there, let me know. And if anyone can do DNA sequencing, also let me know. Yeah, a lot of people find this one, but I think it's pretty much just random. Like I see it in fall a lot more often, but I think it's just because there's not the lower level of herbaceous plant material covering up the infected individuals. I think they stay on, you know, the stick that they're sort of glued to by mycelium for a really long time. Some of these species are like really thinly adhered to the surface that they're on, and the time it takes for the spores to be dispersed is like relatively pretty quick. I'm assuming that this is like a really long-term process. Maybe there is some like pheromone release, so a moth is attracted to this dead moth, tries to mate with it or something, and then leaves and becomes infected. It's just such an open habitat. I don't know how these moths are getting the spores on them. So basically, I don't know anything about this one. It's exciting. And yeah, there's a lot of research out of Thailand, actually. There's this guy, Nigel Hywell-Jones, I believe is his name, and he's published like a lot of work. There's a few people here in the U.S. One of them is Joao Araujo, who I'm working with at New York Botanic Garden. And then, oh boy, there's someone on the West Coast who's doing a lot of research, whose name I will remember later. But there's not many people out there that have access to like full molecular labs and are able to like publish papers at the rate that these guys do, because it just requires a lot of hoops being jumped through, a lot of infrastructure to be set up for you to do that, a lot of funding, and there's only a few labs that are cranking out the majority of the research, which is why it's so important to have community scientists involved. I send a lot of my collected specimens up to New York Botanic Garden, but Joao has like, I've been to his lab and he just has stacks and stacks and stacks of collections. I mean, it's like thousands of collections from all over the world that people are sending him, and he couldn't possibly process it all in his lifetime. So if you're interested in doing the genomic work, if you're interested in typing up the papers, that needs to be done. and it could lead to some really exciting, I don't know. For me, it's about like understanding the ecology of the insect. Like, yeah, we get to name it, we get to see the genetics and compare them to other genetics, but I just want to know, like, why? Why is this happening in the environment? At what rate is it happening in the environment? Where is it happening regionally? Like, yeah, that's the stuff that I want to know. But we definitely need people in all capacities to be studying this. Arinia sepulchralis, impossible to collect. It's always, because it's just so, like, if you know what a slime mold is, a myxomycete, you touch it and it just like disintegrates into nothing. This is a really similar thing. It's a more ephemeral ento, so this is just occurring like in mass underneath rocks. I don't know if you can see this photo, but this is a closer angle, and then this is one of those white dots. So there are like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 white dots, maybe even more up there, under this rock that's sitting overhanging a creek. Like a creek is running directly underneath it. So this is like a really cold, wet, constantly humid, a lot of wind flow in this environment, and you get these epizootic outbreaks. And these are all flies. They're damselflies, they are crane flies, all of these little insects that live their lives in and around the water. This fungus is their predator. And yeah, I've tried to collect it before. It's just like trying to pick up like slime. Yeah, I can't really describe it in a better way. It doesn't dry at all. It just turns into nothing. Like some of these, you'll come up and see it's just like a bunch of insect parts in a box. And it's like, what is this? Well, it used to be a fungus. Some of them preserve really well, but others not so much. Stream dwelling insects, many found in a single location. Yeah, super beautiful though. Because the insect, before you try and touch it and move it, is really well preserved, and a lot of them are really beautiful, hard to photograph otherwise. And then you have the added layer of the fungus. Arthrophaga myriapodina. A lot of people are pretty grossed out by entomopathogenic fungus, and I think they're really beautiful. This is one that I'm like, yeah, I'm a little squeamish about, I guess. But these are so interesting. If you've ever seen a black and gold flatback millipede or a Kentucky flat black and gold millipede, you know, they're very beautiful. They fluoresce under UV light, and they're just sort of like these arthropods that live in the leaf litter. You'll turn over logs or rocks sometimes, and they'll be under there. I had no gauge on how many of these were in the environment until I found this fungus. So you'll find after a heavy rain, you'll go out and usually I'll find them like on skunk cabbage leaves or on little sticks that are about like 3 or 4 feet off the ground. Sometimes I find them at eye level on like tulip poplars, but they just like, I believe it's because once a millipede is infected, it will climb to this point, the fungus will kill it, and then it will trigger a pheromone release in the insect and attract like dozens or hundreds more millipedes to the environment and shoot out the spores through osmotic pressure. And so if you find one, you can find like dozens. I was on a hilltop at the Laurels Preserve last year after a heavy rain with my parents, and yeah, we probably found like 200 of these within 100 yards. And sometimes they'll just be like on top of each other. It's really, really prolific. Yeah, the fungus, you can kind of see it here. I have better pictures on my iNaturalist. You can kind of see it pushing through the chitinous like scales, or like the armor, I call it. Yeah, it infects the greenhouse millipede, which is another invasive species. This little guy, much smaller, but just as common. And yeah, I think I mentioned the spore dispersal is by osmotic pressure. So the body of this insect, it's completely myceliated. It's like a sponge in there with mycelium. And when it rains really heavily, there's all this pressure built up through like the soaking up of water through the sponge. And all of these like little fruiting bodies sort of push out through the armor of the insect, and spores are discharged like hundreds of feet into the environment. Entomophthora muscae is another complex, which I haven't done that much research on. I have a few infected individuals, but if you go out to any agricultural area and you go out to like a wheat field or a corn field and you look at the tops of the corn or you look at the tops of the wheat, you'll just find dozens of flies clinging in like this little death grip to the top of the plants, because it spreads by wind. The spores are dispersed by wind, and I think osmotic pressure too. But some species I'll find under rocks like that. That was the fly that I found under the rock in the first photo in the slideshow. But some of them are in more open areas, which leads me to believe that they're different species. Like, they look pretty different as well. Sometimes, though, this fuzz is like a secondary fungal infection on the entomopathogenic fungus itself. I'll show you a more clear example of that in a second. This is one of my favorites, Jabaliula. I was talking about it before, looking through the beech leaves in fall. But I don't think it's host specific, or it's likely a complex. A lot of different species might need to be delineated out of it if it is host specific. But it's just so colorful. And spiders are already so cool. They're so structural. When they're infected with this, I think they're just that much more sculptural. And yeah, I don't know. I found a few larger individuals infected more recently, but for the most part, this is an American beech leaf. It's about that big. And you can see that little white dot when you look up and the light is cast on the back of the leaf. This is the species that I was talking about that I had seen in North Carolina. It was described by a woman named Kim Fleming, and that's why it received the name Ophiocordyceps kimflemingiae. It infects the workers of carpenter ant species, and there is a specific species that it infects, but I didn't include that on this slide. And it was found in 2009, and this woman is retired, and she just goes out every day with a macro camera to her property in South Carolina and takes hundreds of photos of insects, plants, just everything she can possibly photograph every single day, and uploads them to Flickr in mass. And this researcher, I think from Penn, stumbled across her photos of this species and was like, I don't think this is in PA or in South Carolina. You're gonna have to delete this and re-upload it where you actually saw it. And she was like, you can come down and see. There's thousands of them, on every beech tree. And so we did, and they described it in 2014. And a really interesting characteristic of this fungus is that it forces the ant to bite down on, instead of the midrib of a leaf, the fleshy vein of a leaf, it forces the ant to bite down on a twig, which is obviously going to persist in the environment in a deciduous forest a lot longer than a leaf versus the species in the Amazon, which is a tropical rainforest. The leaves don't fall off of the trees, so the fungus has enough time to complete its life cycle by forcing the ant to bite down on the bottoms of leaves. The further north you go, the forests change more and more, and the fungi that have evolved alongside the ant hosts have not only co-evolved alongside their hosts, but have evolved unique forms of reproduction based on the change and evolution of the forests, which I think is really cool. And this is actually pictured here are 3 different species, and I didn't know that when I made the slideshow. But recently I've been working with NYBG, and we've gotten genetic work back, and what we thought might be 1 new species is 3 new species and an undescribed hyperparasite, which we'll look at in a second. But all of these were found along the Susquehanna River, and all of them were found not biting on the midrib of the leaf, not biting on the twig of the beech tree, but clamped down onto rock underneath boulders that overhang the river, which is a completely new habit. It's really interesting because we basically have the same forests that we have in North Carolina. I mean, there's all this regional ecology that's different, but I want to know what about 6 hours northern difference is so different that this fungus has evolved into a completely new lineage, to a completely different species than its cousins to the south. And once we publish about that and name it, then we can do a lot more research on the habitat. We have permission to collect now, which is really cool. Always get permission to collect. Very important if you're going to publish, because you might have a discovery, but if they say you can't collect, then you never did. And we're really lucky that the Land Conservancy is open to scientific discovery and wanted to work with us. And so, yeah, it's found on the underside of boulders, but also if you've ever looked at the knobby knees of a beech tree, a really old one, there's all these cracks in it right at the base where the roots flare out. And you can also find this species sort of nestled in there, which is really cool. And I believe it's going to be named, instead of Ophiocordyceps, Rowan Nygardii, the species from South Carolina, I think we'll name it Ophiocordyceps campanotus pennsylvanicus, because that's the species of ant that it infects. So, polycephalomyces. I found this and I was like, what is happening? This is some crazy new ento species that I don't even understand, and I brought it into the lab, and you can see that this ant is actually infected with Ophiocordyceps unilateralis, or something in the complex. This black, fungal body is coming out of the back of the head of the ant. So this ant was infected with an entomopathogenic fungus, and then the fungus infecting the ant was infected by another fungus. So the deeper you get into mycology, there's all of these checks and balances in the environment. I'm sure that this has a bacteria or a slime mold that eats it. There's always another hat on the hat on the hat. I thought it was hilarious. It's beautiful, of course, too, but it was sort of shocking to find this. I've only seen this once. There are a few times I've found ants that are infected by this by Ophiocordyceps, and the Ophiocordyceps is covered in this white fuzz, and I've only been able to get good photographs of it a few times, but what I think that is the asexual form of the species, and I just got lucky and saw the sexual reproductive stage, which is likely pretty rare. Ophiocordyceps variabilis. Common when it's common, and I don't really understand fully when that is. I've just sort of like lucked out a few times, and I think that probably has something to do. These are fly larvae infected, so they are minuscule. They're really, really tiny. That's as close as I can get with my camera, and it's like pretty blurry because I don't know the exact size, but this is a log covered in moss, and that is the fungus. So it is just really tiny, and you have to excavate them from the logs, so that's not always the easiest if you don't have the right tools. Sometimes I'll stumble across a log with 20 individuals in the same log. Sometimes I'll just find one, but I don't find them very often. Here, all pictured specimens were found in a single log on the same day. I sat there for like 2 hours and picked apart this log, and every time I turned it, there would be a new one. I said found during summer months after a heavy rain. That's just when I found it. I found it in fall since. So I'm not too sure. I need to find this a few more times before I have like some idea of what is the norm. But really beautiful species. I thought the first time that I found it, it was like some mutated cordyceps, lateris, but pretty different. This is the Ladybug STD that everyone's raving about. Hesperomyces virescens was the name of the complex, and now it's Hesperomyces. Oh, we'll have to look it up at the end. I have a link to the iNaturalist observations. But it has recently been delineated into a new species based on, I think, the species of ladybug. I think these are species-to-species interactions. But really fascinating. These are just little chitinous, fungal. Every single one is a single fungal, I don't even know what to call them, like an organism. But they just sort of take up space on the ladybug's chitin. They're not necessarily digesting it until it's gone. They're not entering the body of the ladybug. I mean, you can see here, like, it's getting pretty serious in this general area. But, so that looks like it might hurt. That looks like there might be a problem there, but...
Is it like a barnacle almost?
It seems so, yeah. They fly with it. I found ladybugs that are like more infected than this, and they were like moving around just fine. I think it's just like a really bad STD. It's like a really bad infection. But also I'll say, almost every single ladybug I've ever found has this on it. So if you find a ladybug, check it out. Bavaria bassiana. Really interesting fungus because it is used in horticulture and agriculture to control coleoptera pests. People spray it as like a biopesticide, which, is better maybe than some pesticides/herbicides, but the question remains, like, is introducing more of this fungus, no matter if it's like endemic to the environment, beneficial to the environment because it's so non-specific in what it infects. So like all of your native beetles are also going to be infected by those spores that you're introducing into the environment. So cool in closed systems if you have a closed system, which not many people do. Called sugar icing fungus, one of the few fungi in this group that I'm presenting on that have a common name. I would like to see some people inventing common names. Let's get creative with it. I want to get more bulrush caterpillar common names. Like, I love common names. I think we should use more. Unpopular, yeah. People think regionally. Like, I think it really brings out like cultural differences. I think we should, of course, in the literature rely on scientific names. But sorry, unpopular opinion. Yeah, easily spread by wind currents, not host specific. Yada yada yada. These are entomopathogenic fungi that I know nothing about. I don't know what they are. I've been unable to tell. Hopefully I have gotten a lot of these submitted to people with molecular labs, but again, those people are few and far between that are working with these groups. And even if they do get the genome, sometimes these are just so new, quote unquote, that there is nothing. They're like, yep, it's new, and that's it. But let's just quickly go. We'll talk about resources in just a minute. See if this works. Because since I last updated this slideshow, I have found quite a few others. And at the very least, there's something pretty to look at. Whoops. Don't look, don't look. Dun, dun, dun. Ooh, this was a cool one. Okay, that's probably Hypocreales. Really, really cool.
Is everything being talked about today in
An ascomycete? Yeah, I think so.
Is all of it in
What was that?
is all of it in the Sorgheriomycetes?
I don't know, actually. We can find out. Bada bada ba. It looks like it for the most part, yeah. I'm not as good at taxonomy as I am at fieldwork, I'll say. But, ooh. But thank you for asking. Okay. This guy, a Phyrocordyceps dipterigena, I found in Tennessee when I was there. This is a fly without its head. So like the head has come off, and these are the other two sections of the body, and the wings are off. I went on a mycology hike along this trail in the Great Smoky Mountains, and I was like determined to find some entomopathogens, and I went down to the creek, and within like 5 minutes of looking, just found this fungus. And that's another reason that I'm really excited to explore like the rainforest down there, because I was down there for like a day, and I think the biodiversity or the microdiversity is really, really high. It's just there's not many people that know where to look. Slideshow. This is a spider. Those are the eyes. If you can see my cursor. This is crazy. I was taking my dog on a hike. I was walking my dog. Sorry. Very tired today. I was walking my dog, and we were looking. She was really complaining that I was stopping so much to look at the leaf litter. But I scooped up an oak leaf, and if you've ever seen one of these insects before, they are minuscule. They're like the size of a hair. They're just really, really tiny, really hard to see without a hand lens or a camera. And I didn't see it. There were some butterfly eggs on this leaf that I wanted to look at under the microscope, and I brought them home, and I found this on it. And I've seen this insect before, but it just had like very clear fungal growths coming out of it. And this is, again, something I've sent up to NYBG that Joelle looked at and was just immediately like, yep, that's a new species. And this was right behind my house, like in the creek behind my residential neighborhood house. I mean, like, they're everywhere. We could probably go outside the library and find a new species right now. Maybe we should. And this was found at the last MycoFest, which is a mycological festival in Pennsylvania that's held every year. And I'm not sure about the ID of this, but it was growing out of this little Coleoptera larva. And I've never seen anything like it again. I think it's a Phyrocordyceps stylophora. And, well, I'm not sure though. I didn't collect it, so we'll never know. And that's that. That's everything. Oh, wait, no, it's not. Just kidding. Resource list, which I can share with all of you afterwards. Oh yeah, and we should have some time for questions. iNaturalist, Facebook group, Cordyceps, Elaphocordyceps, Metacordyceps, Phyrocordyceps, Clavitypes. My Instagram, Rowan Nygard. I post a lot of photos on there. I talk a lot more coherently when I have time to like write something up on there. That third link, the third or fourth bulletin, is a really, really cool website by Shigeo Atake, who is a Japanese painter who is like a surrealist painter. He's a contemporary artist, and he just hyper-focuses on like Cordycepitacean subject matter, and his website is like Alice in Wonderland. You go in there and you get lost, and you don't even know how to get off of the internet again. It's like, it pulls you in. It's incredible. I highly recommend it. Vegetable wasps and plant worms, 1892, parasites and social insects, more accessible and affordable than the previous book, 1998. The Cordyceps Cultivation Handbook, if you're into like the edibility of cordyceps, and the really interesting like modern history of their cultivation in the U.S. Atlas of Invertebrate Pathogenic Fungi of Thailand, like I was talking about earlier. Nigel Hywell-Jones does just such great work over in Thailand on entomopathogenic fungi. So I think that like a lot of the work we're doing here in the U.S. to describe new species, it's great to have like a backbone of work to look at in another country, because those species have evolved from a common ancestor a lot of the time, and we can look at the genetics there. And yeah, I'm waiting to buy a copy of all 4 of those volumes, but they're very expensive and you have to import them, so that's it. Any questions? Thank you.